Good morning, everybody. This is Schang. I'm a professor of chemical virology in the Baker Institute for Animal Health. So I have been involved with the antiviral field for this long, and I have been following the outbreak since basically it started, as far as we know, in China in late November, early December last year. You may have attended some of the webinars that we gave a couple of months ago, providing updates relevant to veterinarians or anybody for that matters with respect to SARS-CoV-2 and the COVID-19 pandemic. Since then, there have been a few updates and improvements with antiviral therapy. At the moment when the webinars were being held, several potential compounds were being explored, some of them which had a good probability of being successful as based on the expertise on antiviral research and development that exists in the world. One of them is remdesivir, which by now has been approved by the FDA for use against COVID-19 or SARS-CoV-2, and it has proven to be effective and helpful, albeit it's not the magic bullet. It doesn't cure everybody, and it's not going to solve the problem, but it's an effective drug that is being used at the moment in the clinics with success. At the moment, there was still some discussion about chloroquine and hydrochloroquine. Both of them have now been disapproved. The approval has been removed by the FDA, and even the large-scale clinical trials that were going on around the world have all been halted because chloroquine and hydrochloroquine have been concluded not to be active and to have the non-toxicities as any drug does. So the benefits don't outweigh the risks in this case. There is also a major development, not in the antiviral field, but in the management of the disease. And there was quite some, not controversy, but it was complicated to understand what was the role of anti-inflammatory therapy in the management of the disease. By now, it has been very well established that proper use of corticoids in patients that are in the hospital is extremely helpful and has a very positive benefit. So there had been significant upgrades in the treatment of the disease and the viral replication with remdesivir and judicious use of corticoids being the standard of care these days. So although we do have the first antiviral that is active against SARS-CoV-2, this drug remdesivir is definitely not the final answer and is not fully effective. So antiviral research continues at really, really frantic pace. There are many, many different projects that are being pursued all over the world. And one thing that is actually quite exciting and it raises the enthusiasm is the level of sharing of information across antiviral researchers. Probably the next drugs that are going to get into clinical trials against SARS-CoV-2 are actually not so new drugs. They're nucleoside derivatives that were developed many years ago. One is called NHC, and the other one is a prodrug of NHC, basically a molecule that gets processed in vivo producing NHC, which is called EIDD 2801. I apologize. We as researchers use code numbers and letters when we are developing things early on. Eventually they get a better name like remdesivir. So these two drugs have proven to be active against SARS-CoV-2 in culture. And because the drugs were pre-existing and they were being tested against other viruses, we know that they are active in animal models against a variety of respiratory viruses, including flu and including the original SARS, what is called now SARS-CoV-1, and MERS, which are both other human pathogenic coronaviruses. So these drugs have been announced to be entering to phase 1 clinical trials, which is safety only. Both of them are nucleoside derivatives. They are somewhat related to remdesivir. Their mechanism of action, however, the way that they inhibit the replication of the viral genome is different from that of remdesivir. In fact, NHC or EIDD 2801 do not inhibit the replication, but make the enzyme that replicates the viral genome make too many mistakes. So the replicated genomes are defective, and in the end, the viral infectivity is lost. There is also another group of molecules which is also based on nucleosides, and I should emphasize that nucleosides are one of the most common structures in antiviral drugs, the other one being protease inhibitors. There are another family of nucleosides which are called fleximers, with which we are working in collaboration with Dr. Kathy Radke in the University of Maryland, Baltimore. And these molecules, again, have been shown to be active against SARS-CoV-1. They have been shown to be active against MERS, and they have been shown to be well tolerated in animals. We're now working with some of these compounds with SARS-CoV-2, and we'll see how fast we can progress with these other molecules. The other big family are the protease inhibitors that I mentioned already. And this is a platform that has been developed quite extensively. The first protease inhibitors were developed against HIV back in the late 80s, early 90s. And since the first ones, the anti-HIV protease inhibitors have been refined through several generations. And when another virus came of importance, hepatitis C virus, that also has a protease, the same technology was applied to develop protease inhibitors active against hepatitis C virus, and those drugs also have been very successful and they're in clinical use. These days, Hep C is actually a curable disease in a little bit more than a month or less than a month for most patients. So these type of molecules are now being developed against SARS-CoV-2. The crystal structure of the proteases are known, so that facilitates the design, and there is a very significant effort and investment put into the development of protease inhibitors. There are other molecules being developed against a variety of other targets, but probably the two that are going to get to the clinics first are inhibitors of the viral polymerase and the protease, because those are very mature platforms that are very well developed, that are now applying all the knowledge towards a specific virus. Part of this worldwide effort in trying to develop new drugs against SARS-CoV-2, as I mentioned before, we have gotten to screen all the different compounds that we have against. First, we use an endemic human coronavirus, which is called OC43, because then we can screen large number of molecules and biosafety level 2. The molecules that are found active then are moved into a biosafety level 3, and we're testing them against SARS-CoV-2 itself. We are working with some molecules that we have been working for many years. We're working with Dr. Kathy Radke with the fleximers. We are working with Dr. Fred West with a specific family of compounds. And we're working with a variety and a number of other chemists in the US, Europe, and Canada with different types of molecules. The molecules that are shown active and safe will then be advanced into animal testing. And then we'll see what happens.